In this session, our next speaker is Neal Swerdlow. He is a professor of psychiatry. He's currently the vice chair for education and training. He is the director of the psychiatry research track, which he worked his tail off to have renewed, and it is just a very effective program. I'm very, very proud of him. As well as, for years, he was the director of education for medical students, psychiatric medication for medical students. We were fortunate to have him join our faculty in 1991, and his levels of expertise certainly go beyond issues related to schizophrenia. He has done some stellar clinical as well as basic research and basic applied to clinical for two very difficult to treat disorders. One, very difficult to treat when at its extreme, obsessive compulsive disorder. And two, and of course there's some level of overlap regarding these, Huntington's disease. Today's talk the lecture for his talk will be shown by him, so that Neal Swerdlow. The title that we have in the program is just a little bit off, so he is going to present his own title. Neal Swerdlow.
I want to thank the committee for allowing me to tell you a little bit about the neural circuitry of mental illness as informed by models of sensorimotor gating. I'll start with some background, tell you about CSPT circuitry, some more about cross-species models, a theme for this morning, and then what's happening in the future. So somebody smart said that research careers are equal parts bad ideas, good ideas, and luck. That's certainly been the case for me. Mark Geyer told you that weak sensory events inhibit motor reflexes. We've known about this since the 19th century. Ivan Sechenov, a Russian physiologist, showed that tactile pre-stimulation can inhibit the pain withdrawal reflex in humans. Helen Peake, a postdoc at Yale, used this gizmo to swing a wooden spoon at Yale undergraduates' faces and then inhibited the blink response with a weak click to the ear. Hoffman and Fleshler published a paper in Science magazine with six rats, Science magazine. On the y-axis is startle magnitude to an intense noise burst, and you see that the startle response completely disappeared when it was superimposed over a background of weak 1 per second sound pulses. And Fran Graham summarized this in her 1974 presidential address to the Society of Psychophysiology. And had I known about any of these things, I would never have had my first really bad idea in 1978, which was to use pulsating tail pressure as a stressor to potentiate the startle reflex and then use that to screen anxiolytic drugs which would reduce the stress response. And much to my chagrin, in the first experiment that I did, I showed that pulsating tail pressure actually cut the startle reflex here on the y-axis amplitude by half. I had absolutely no idea why this happened. I had no explanation. But in a stroke of good luck, one of the 3 journals that we got at the Amherst College library was Experimental Neurology. And that same year, Jan Stevens published a paper showing the effects of photic stimulation on neural activity, activating expected areas within the visual system, but also this obscure basal forebrain nucleus called the nucleus accumbens. Like some people in the audience here, I learned about the nucleus accumbens from Jan Stevens' 1973 Anatomy of Schizophrenia article, where she described the nucleus accumbens as the limbic striatal gate and proposed that dopamine hyperactivity in this area might be responsible for some of the symptoms of schizophrenia. Well, I knew where the nucleus accumbens was because it was in the Pellegrino and Cushman rat atlas, and I could turn to that page, dial in the coordinates, and make radiofrequency lesions of the nucleus accumbens shown here. And when I did that, the effects of these tail pinches, pulsating tail pressure on the startle reflex went away. And as I was writing up the discussion section for my thesis, I came across this paper by Braff and Geyer in 1978, and I've actually replotted their data here with bar graphs to show you the similarity in what they found and what I saw in our lesioned rats. On the y-axis, startle magnitude, and what they showed was that the reflex inhibiting effects of weak prepulses were diminished here in a schizophrenia patient compared to what they were in healthy control subjects. And 38 years later, I can tell you that the inhibition of startle by prepulses is impaired in a group of brain disorders. In some cases, it's linked to functional deficits in those disorders. It's used in animal models to predict therapeutics. Mark's shown some of that work. It's regulated by identifiable forebrain circuitry with a prominent role and heritable role for dopamine in the nucleus accumbens, and it's associated with the expression of certain genes within that nucleus. But before those 38 years, I had to get from Amherst, Massachusetts, driving across country to land at the Salk Institute to work as George Koob's first graduate student. And here's George. Everybody knows George in this audience, I'm sure. There's me standing behind him. And my work in George's lab was on the ventral striatal palatal, that's another word for the nucleus accumbens into ventral pallidum circuitry, using a dopamine supersensitivity model where we achieved using 6-hydroxydopamine to produce denervation supersensitivity in the nucleus accumbens, studying locomotor activity. But what I really wanted to do was to use that supersensitive dopamine model to study pre-pulse inhibition of startle. The problem was that George had locomotor boxes, but not startle boxes. So I went about trying to build my first startle box. And late one night as George was leaving, he looked at me and said, "Swerdlow, don't Braff and Geyer have a bunch of those things?" And I said, "Braff and Geyer, they wrote that paper." He said, yeah, I think they're doing this stuff. I said, "Well, where are they?" He said, "They're in Hillcrest." So that was my first stroke of luck here as a UCSD student, and that was finding Mark and David, who were indeed in Hillcrest, and Mark indeed had a bunch of those startle boxes that I didn't have to build. And together we applied the dopamine supersensitivity model to pre-pulse inhibition measures in rats and published his first findings in 1986, which were a bridge between Jan Stevens and others' models about mesolimbic dopamine overactivity and the work that Mark and David had been doing on information processing and sensorimotor gating. And so this was the first connection of those two models. I then set about trying to understand how dopamine overactivity in this ventral striatum area made its way through brain circuitry all the way down into Pontine's circuitry that regulated pre-pulse inhibition, and ultimately the spinal circuitry that generated the startle reflex. The only problem was I knew very little about brain circuitry, and that was the next stroke of luck, which was in 1983. I took a neurophysiology course with this man, Phil Groves, one of the great teachers in our department, and certainly a cornerstone of what we've understood about neurophysiology, particularly within the basal ganglia. And the final exam paper I had for Phil's class that year was to compare Phil's model, which came out that year, of gating mechanisms within the striatum, which was based on this lateral inhibitory matrix of the medium spiny 1 output neurons in the striatum, gating or filtering information that was coming down from cortex and thalamus as it was involved in the regulation of movement. To compare that circuit model to one that had also come out that year in the Annual Review of Neurosciences from Penny and Young. And their model was a 3-loop model that involved connections between cortex, striatum, pallidum, and thalamus. Their model provided an iterative mechanism whereby subcortical activity within loop C could then modify activity within this corticostriatal positive feedback loop, which was a way of essentially shifting or altering the stream of positive feedback between cortex and thalamus. And I, thinking about that, started to think about how that same process in parallel might be working in terms of not just regulation of movements, but the regulations of thought and affective states. And that iterative component I thought was particularly valuable for that. So I liked their model better. But the problem was that Phil was grading my exam. And so that led to the next important good idea that I had in my career, which was to steal Phil's medium spiny lateral inhibitory matrix model and to stick it into the Penny and Young's 3-loop model, and then came up with a model which involved these same 4 nodes, corticostriatal pallidothalamic circuitry, but moved them up into portions of the brain that involved limbic activity rather than simply the motor cortex. Still the 4 same general components, but most importantly stuck into it, in the middle of it, Phil's lateral inhibitory matrix, which made the whole thing work. It was sort of like a critical component of the machinery. We could then model the dynamic changes that would occur within every level of the circuitry if we just shifted, for example, the amount of dopamine within the nucleus accumbens and use this to account for a variety of different psychiatric states. And this model, like everyone before it and everyone that will come after it, was wrong. It's been recreated in several different forms. This is Jonathan Mink's form. Again, the CSPT nodes remain a critical part of it, and indeed, many of the disorders that I've spent my life studying are housed within the circuitry. Huntington's within the striatum, Tourette syndrome, which I've spent a good deal of my career on, within cortical and striatal components, as is OCD, and certainly schizophrenia at many different levels of this circuitry. And working down in Hillcrest, a lot of it with Mark and David, we then went and tested whether those disorders, which are known to be housed within this circuitry, have abnormalities within gating function. And indeed, the first study we came out in '95 in Huntington's patients, on the y-axis here is pre-pulse inhibition. I won't keep repeating that. Compared to matched control subjects, Huntington's patients are profoundly deficient in sensorimotor gating. Actually, Javi Castellanos first reported the deficits in Tourette patients. We replicated that and extended it into other groups, and others have replicated it since. OCD patients we reported in '93 are deficient in sensorimotor gating. This has been replicated by a number of other groups. Susan Amari's group most recently showed that this deficit could be normalized with deep brain stimulation in the nucleus accumbens, connecting that to our circuit models. And certainly we've spent a lot of time sort of doing a deep dive into the deficits in schizophrenia patients, picking up on David and Mark's original work, which has been replicated more than 40 times. And we've looked at things like the relationship between these deficits in pre-pulse inhibition and function, here shown on with GAF scores, showing that the more impaired they are, the worse gating they have, and vice versa. But we've also looked at the relationships with antipsychotics, typicals and atypicals, as well as nicotine, sex differences, and a lot of other variables that have factored into the gene finding studies that David and the COGS have done. So much of what we've done over the last 20 years has been to leverage the biology of pre-pulse inhibition to study a number of questions of interest as they relate to psychopathology and to normal brain function. I won't have a chance to even touch on most of these. Many of you in the audience here I've collaborated with to study these various different aspects of mental illness. I'll focus on neural circuitry, and this is sort of just a summary of, sort of a cross-section of the types of work that we did, very old school, I will say, anatomical and neuropharmacological manipulations. You can never get a grant for doing these kinds of things anymore without DREADS or optogenetics or anything, but pretty much mapping out the circuitry starting in prefrontal cortex, going all the way down into the pontine tegmentum to understand the brain mechanisms that accounted for how activation within at the dopamine receptor in the basal forebrain could ultimately influence the circuitry. Along the way, we found a number of other interesting things, one of which is that the sensitivity to dopamine within the system is easily heritable within rats. This is work that Paul Schilling helped us do, that we could breed it in and out. Here at the albino spragues, showing a tremendous sensitivity to a very low dose of apomorphine disrupting gating. These hooded Long Evans animals are very insensitive. We can then cross them to produce the F1 and then back cross to the N2 and show the graded effects of this heritable pattern of dopamine agonist sensitivity, which we've chased down in a number of ways. Paul helped us identify a number of gene expression differences within the nucleus accumbens that accounted for this, and particularly robust differences within catechol-O-methyltransferase. So, 38 years later, I can tell you that the inhibition of startle by prepulses is impaired in a group of brain disorders, in some cases linked to functional deficits. We can use it in animal models to predict therapeutics. It's regulated by an identifiable forebrain circuitry with a prominent and heritable role for dopamine within the nucleus accumbens, and it's associated with the expression of certain genes within that nucleus. So what's next? We're working on two ideas. At this point, I can't tell you if they're bad ideas or good ideas. That's something we hopefully will know in the near future. One is that can we use prepulse inhibition as a biomarker to predict treatment sensitivity? The other is something we call PACT, for pharmacologic augmentation of cognitive training. And I'll tell you a little bit about both of those, but the one really good idea I had of late and last 10 years was to work with this gentleman, Greg Light, who really has helped advance these ideas, and we've had a great partnership in being able to work on this science together. The biomarker idea is a relatively simple one. You know that if we use clinical phenotypes as the basis for picking a medication, they're highly heterogeneous. We apply an intervention to a heterogeneous population. Even if there's a few sensitive people within there, most are non-responders, and this results in a treatment failure. On the other hand, what we would like to get to is to identify that sensitive group upfront using a biomarker, apply the intervention, and that will produce a treatment success. The question is whether we indeed can do this. These are data. Each one of these dots is a schizophrenia patient. On the y-axis here is the magnitude of a drug effect. This drug happens to be memantine, but I'll tell you we're working on a number of drugs. And this is pre-pulse inhibition and a number of other measures that Greg works with, all of which involve early auditory information processing. And so you see here that there's some individuals who show an increase in pre-pulse inhibition with this drug. We call those people drug sensitive. Others that show no change, we call them drug insensitive. And the question we're asking is whether this phenotype or biomarker predicts whether those individuals are going to be sensitive or insensitive to the drug. We've got clinical trials in progress now where we're doing that. Like I said, it's too soon to know if it's a good idea or a bad idea. The second new idea we're working on is called PACT, Pharmacologic Augmentation of Cognitive Training. The general idea here is that the old paradigm was, particularly as it relates to schizophrenia, is to find what's broken and try to fix it. The new paradigm is to find what still works and to strengthen it in the service of targeted cognitive therapy. We have some early proof of concept data on this using pharmacology in schizophrenia patients, in this case using the psychostimulant amphetamine to enhance sensory training within these individuals. Essentially, schizophrenia patients learn more if you give them an acute single dose of 10 milligrams of amphetamine, and then when you test them a week later, they've retained more. These are in antipsychotic medicated schizophrenia patients, so it's not a state-dependent phenomenon. Again, we don't know if this is a good idea or a bad idea. A great idea has been for me to be able to work with all of these individuals. I'm sure many of them are out in the audience here. Others are dispersed across the lands advancing science and medicine. The best idea and the luckiest thing I ever did was meeting my wife here at UCSD in 1981 and being able to raise a family with her. Nancy Downs, also a professor of psychiatry within our department. She runs the college mental health program. And with that, I'll stop and thank you for your attention.
Another superb talk. So while we're in the midst of learning something about schizophrenia and schizophrenia research, we're learning something also about human beings and overall approaches to science. We've learned translation is incredibly important. We learned that consortia can develop incredible ideas that allow you to do such things as look at specific endophenotypes, the smaller ones, and how they relate to specific areas of genes. We just learned some superb science, step leading to step leading to step. But what I would like to emphasize most is that indeed there are good ideas, and from your talk, it's pretty obvious that the good ideas are seldom completely right. And then there are bad ideas that actually can lead to some incredibly good ideas, especially if you look for what it is that you found that was the opposite or that's different than what you were looking for in the first place. And then we've also learned luck. And luck is an incredible part of what we do, because if you think of it, how many people have awesome ideas with as good an evidence as you might have for your ideas, but as they follow them through, by bad luck, it turns out to be not exactly what they thought it would be, if anything at all. And by good luck, yours, but it's often by paying attention to when you have something wrong, what is it that you can learn from it?